Well, thank you very much, Silke. It's a pleasure to be opening this meeting, indeed. And I'm going to try and give you a bit of an overview of my journey since I was about your age as a new generation scientist to I've been here 29 years in September. So it's a kind of quick swan through 29 years to give you an overview of what we've been doing. Now, I always think it's really valuable meetings like this because it's when you meet people and you talk to people and you just a sort of kernel of an idea comes up in your mind that you can then mature. And it was that sort of meeting and discussing with people early in my career that got me thinking about the sort of feature of this picture. And it's like, why is it that all the plants of the same species flower at roughly the same time? And it was something that, a comment someone made to me. And and then I had at home when I was a teenager, some cherry trees in the backyard, and they all would flower about the same time. And it sort of occurred to me that, this was really quite an interesting question. And when I had my own group, this was the kind of thing I wanted to look at. Now, if we think about it, plants of the same varieties, anyway, flower about the same time, but different plants flower at different times. So the actual flowering time is very plastic indeed. It responds enormously to the environment, whether it's temperature or photoperiod, various environmental cues. But plants, and the example shown here is just one. This is the same genotype. Get the right button here. But it's just grown in different conditions. So the same genotype can flower very differently, very plastic response. But of course, it has to get that timing right to flower at the right time for reproductive success. If not, it's kind of toast. So flowering time is a hugely important trait for reproductive success in plants. And at the time, when I came, Arabidopsis was just a kernel of an idea. There was a couple of labs in the US pushing it hard. There was no genome sequence. There was no stock center. There was no physical map or nice mutant collection or anything. But the idea out there was that you could use molecular genetic approaches to dissect these really complicated problems and then identify the genes involved. That was really new in the plant world at that point in the late 80s. But when I first arrived, a group of us, a colleague here and then others collaboratively throughout Europe, said, well, let's all work together and look at flowering time. We chose Arabidopsis, we chose a genetic system. And, this is the very simple summary of a lot of people's work over the first 6 or 7 years. It was actually profound in that what we realized was that all these different cues that affect flowering, and there are environmental cues, there are endogenous cues, somehow all of these play a role in flowering, but no one quite knew how they all fitted together. They converged to regulate the same set of target genes. Now, when we draw a picture like this, actually that was quite influential in terms of thinking of flowering in species other than Arabidopsis. Because if you take rice, it needs short days. If you take chrysanthemums, they need short days. Whereas if you take brassicas, they need winter. If you take maize, it doesn't seem to have much response to environmental cues. It's endogenous signals that are important. And no one quite knew how these very different physiological effects could be functioning at the molecular level. But a schematic like this said, well, they could all be functioning in a rather simple way in that all of these different inputs, which are differentially important in different plant species, will come down and regulate the same set of targets. And when we first proposed this, we're sort of treating with slight skepticism, but I think in the end, this is the way the thinking has evolved, is that indeed there's a set of genes in most plant species that are required to be switched on for the meristem to switch to a floral fate. And you regulate the activation of these genes, lots of them, very complicated, lots of feedbacks, but this is the simple, reasonably sort of intuitive way of thinking about flowering time control generally in most plants. Now, so there were various different cues, and lots of people have followed different ones. The one we followed was winter. And people think, I'm going to tell you about some field experiments. And of course, we do winter, so we look at extremes of winter, and we go up to northern Sweden. And I remember my colleague saying to me, you made a big mistake at that point, Caroline. If you'd done photoperiod, you could have gone somewhere in the equator every year. But anyway, we chose winter. We chose winter, actually, again, one of those slightly odd things that happened to me. I was a postdoc in California, and I went to a shop and I bought some tulips. thought I was a bit homesick. I'd have some tulips to make me feel a bit better. And the person in the shop said to me, now put them in the fridge for 6 weeks before you plant them. And I thought, wow, this is really weird. These plants actually need, these bulbs need 6 weeks of cold. They actually need the cold. It's not just that, they don't grow very fast or over winter. They actually need the winter to do what they need to do. And that's what got me into vernalization, into this requirement for winter. And winter, of course, is very interesting. It's not just, we stick the Arabidopsis in the cold room for 6 weeks or something. Well, that's not at all what the plant experiences. Here's a typical, from northern Sweden, temperature profile over these period. This is sensors now on the ground next to the plant. These plants are experiencing massive fluctuations in temperature over these long time scales, and somehow they're having to read what is going on in these very noisy signals to work out, has autumn finished? Is it now time? Has winter gone by? Is it now time to flower? And of course, what they need to do is make sure that they don't get confused by a cold snap in autumn, because then they'll flower, as in my plant before, in the middle of winter, and the reproductive success will be very poor. So that's what we've been studying, this whole, how does winter, and if I just go back, how does winter influence these floral pathway integrated genes? And they work via the winter works via a specific floral repressor, which I'll talk a lot about, and that keeps all of these genes off, and then winter switches this off, allowing these genes to be expressed. Now, the temperature doesn't actually cause flowering, it just allows these genes to be expressed when these other environments then come on board. So we asked 3 questions in the lab when I first arrived. We thought, okay, we'll look at some varieties need to overwinter, winter annual types. Think about crops, winter wheat, spring wheat. Winter wheat you have to put in the ground in the autumn, overwinters and then flowers. So why do some need winter and some varieties don't? If you need winter as a plant, how do you register these complex temperature profiles? That was the second question. And the third one was, because winter in different places is very different. So how do plants adapt to different winters? Now, at the time, as I said, Arabidopsis was just an emerging model system, reference plant, as we should now call it. But there'd been a lot of genetics in crops because, of course, vernalization and this winter-spring variety type is hugely important for a lot of different crops. The breeding of winter and spring varieties has extended geographical range of many of our major crops enormously. And I'll get back to this at the end, and I think this will fit in nicely, hopefully, with the speaker later on, because as our winters change and our varieties now become not so adapted to the conditions we've bred them for, we're going to have to understand the molecular basis of vernalization. But we didn't want to do the genetics in the crops, so we basically turned to Arabidopsis as this genetic system to tackle what was involved. Now, if any of you have seen Arabidopsis, and I imagine most in the audience have, you probably sort of have this image in your mind of this rapid cycling type of plant. That's why, of course, we all use Arabidopsis for rapid genetics. But in fact, if you take 100 varieties, especially from around here, and put them in your long day, nice warm growth chamber, about 70% of them will look like this. They are winter annual types. So not the Arabidopsis you're used to in your experiments at all. They are very vernalization requiring, whereas if you give them vernalization as a seed, 6 weeks of cold, they will then look like this. Now, I'm not going to go through all of the early work, but it became very clear that there was one major determinant for this vernalization requirement and for actually the ability for cold to make it flower early like this, and that's this gene, flowering locus C. It's one of those names that I wish I could change, but it was already called flowering locus C, and that's what we've sort of been stuck with. But just think of it as a brake. If you're expressing this protein, you block the ability of the meristem to switch on all the genes required for a floral fate. So this brake keeps flowering off. The plants get bigger and bigger, making more and more leaves. And actually, there's a lot of quantitative regulation of FLC to determine how much winter is required. And then the prolonged cold comes along and switches FLC off. So this brake is a MADS box transcriptional repressor, as I said, preventing activation of a whole suite of genes. Now, we've worked on this for many, many years, and our 3 questions that we started off with, why do some varieties need vernalization and some do not, comes down to the transcriptional regulation of FLC and if it's high or not. And, I say to people, it's the major determinant. It's probably affecting about 80% of the phenotypic variation is due to expression of this one gene, which is really quite remarkable. Because when you mutate many genes in the genome, there's probably 150 genes you can mutate and get a flowering phenotype. But natural variation seems to have focused on selecting variation at this one locus. So this is true for Arabidopsis. We've now got work in a lot of the Brassicaceae, the cabbages, the cauliflowers, the oilseed rape. Exactly the same story, happily. It's just that there are many more copies in those genomes. So this is true for the whole of Brassicaceae, but what I'll talk about today is in Arabidopsis. And I'm going to focus on this bit here, the prolonged cold, because the title is Sensing and Remembering Winter. So this is the bit I'm going to talk about, the switching FLC off in response to cold. Now, what was clear early on, and many different groups studied this initially, was that when you give the plant cold, and here's our plant with no cold making lots of FLC and flowering very late, but with increasing cold, and this is weeks, you slowly but surely switch off the expression of this break and therefore allow flowering to occur. And the intriguing thing that people had noticed for a long time ago was that if you take plants out after a certain amount, then the expression will remain at that level throughout the rest of development. And those plants will flower a little bit later than plants having given 4 weeks. So there was a sort of quantitativeness to this silencing that people didn't understand. And that was really an important part of what I'll be telling you about. And the other thing is that, as you take the plants out and put them back into the warm, the gene stayed off. So it wasn't that just it needed warm or it needed cold to switch it off. The cold switches it off, and then it stays off in the subsequent warmth. So that's shown schematically here. Here's an expression of the gene in autumn. It goes off very slowly, as I showed you, and then stays off. And this is this epigenetic silencing. Now, epigenetics has become quite a kind of hot topic at the moment, but it's always interesting to me how many people have come across it. So let me ask you, who of you could explain to me the basis of epigenetics, just so I know how many people in the audience? Okay, a smallish number. Okay, just it's always very useful because, there's a diverse audience and it's not quite clear. So this is basically now epigenetic silencing of a gene over weeks. This is the whole of spring. So the gene is switched off, and then whatever switched it off is remembered through many, many cell divisions. And that's really the basis of epigenetic regulation. It's this instruction by one gene affecting one gene or several other genes, instruction by one regulator, I should say, which is then remembered by a different system, by an epigenetic memory system that keeps that expression state through cell division. So that different cells of a plant can be in different epigenetic states, so that each cell can receive an instruction and then keep that instruction. Now, what I'm going to do is tell you a little bit about that epigenetic instruction, the remembering winter. So that's in my title. And then I'm going to go back. So this we've published over many years now. And I'm going to talk about, at the end, slightly more recent work, because now we're turned to sensing winter. So, okay, let's think about remembering winter as the first bit. And as I said, the mutants and what we got into through this epigenetic regulation, through this vernalization, was epigenetics. And what we did was to take plants that needed vernalization to vernalize, but also to mutagenize, and look for the ones that no longer could respond. That identified mutants, we call Vern mutants, and then we went in and cloned the genes basically defined by these Vern mutations. And we got into the world of Polycomb, epigenetics. Now, this is the beauty of forward genetics. You have no idea where you're going to end up. So you then go to the literature and you think, okay, Polycomb, what do I find out? What's going on? Now, it's a terrible name. Again, they were landed with a bit like FLC, Polycomb. It turned out to be in Drosophila. People were looking for mutants that behave differently in terms of their body plan. So this mutant had more combs on its head. The males have more combs. And so the mutant happened to be called Polycomb. And that was the first mutation in something that's now been clearly identified as a very conserved chromatin mechanism, that mediates this memory mechanism in most high eukaryotes. So everybody talks about Polycomb and then forgets to ask, why on earth is it called Polycomb? Anyway, the name matters, unfortunately. So it's this well-conserved chromatin silencing complex. So that was one set of mutations that came out. And the others were some PhD proteins, which I'll get back to, which these are their names. But these are, one of which is cold-induced. And they're very important to sort of activate this Polycomb complex. Now, I'm just going to do a sojourn into a bit of background of epigenetics, just so that we're all on the same page here. And so if you went to the literature, as I say, to sort of understand what on earth this Polycomb is, or epigenetics in general, most of it involves chromatin regulation. Not all, but most of it, and certainly the literature is full of that. So if we look at our chromatin, we unwind our chromosome into the DNA, into the chromatin, I should say, and then unwind it even further. The DNA is wrapped around the histone optimers, the nucleosomes, and these nucleosomes are made up of histones with the tails, the amino-terminal tails, sticking out. It's these little blue things here. And they are post-translationally modified. The other thing that's modified is actually the DNA sequence itself, the DNA methylation, for example. So you have modification of the DNA, either on the protein or on the DNA itself, which changes the expression of that DNA sequence. And you can think of it a bit like a computer. So if the gene is the hardware, well, the gene has no instructions as to where to switch on, when to keep off, and all of those things. So that the software, the information layered on top of that hardware, is then the epigenetic modifications giving that gene the instructions. And if you look actually at Polycomb, at DNA methylation, they are very widespread, very conserved, most eukaryotes, from either defining which cell, the fate of the cell, from stem cells in your body to the various forms of the cells and making up mammals, to the differences, of course, between identical twins is an epigenetic modifications. And then actually now when people begin to look at the basis of many cancers, they find that a lot of the genes that seem to be causative for cancers are influencing methylation of very specific and important targets, like the BRCA1 gene for breast cancer is about hypermethylation of BRCA1 in many cancers. So epigenetics seems to come up, or misregulation of genes due to misregulation of epigenetic processes seem to be very important in many systems. The other very famous example, of course, is the cat. How can I give a seminar without showing a picture of a cat? Everyone goes, ah. But actually, the tabby cat is a classic example of an epigenetic phenomenon in that the orange color gene is on the X chromosome. And of course, all of you in the audience who are female have got two X's, one of which shuts down early in embryo development. And that's random. Which one gets shut down is random in each cell. So you basically get, depending on which is shut down, patches which are going to express the color gene or not. And of course, as the embryo then develops, these patches get bigger and bigger, and you see these reflection here of epigenetically modified orange gene, which is then different from this patch where this X is shut down. So you can see that for the same gene, you can have different epigenetic modifications within the same organism. So if I go back to this schematic here, what we then came up with in our mutants was that we were somehow involved in this Polycomb silencing, which is actually very focused on modification of one of these tails to make lysine at the histone 27 position methylated. So it adds 3 methylation marks, so it's trimethylation at histone 3, lysine 27, K27. So you'll see in lots of the papers this sort of alphabety, spaghetti sort of histone modification mark. Here you've got histone 3, lysine 36 has trimethylation, and so on. And so that's a lot of the basis is differential methylation of the different positions on the histone tail are the software instructing which protein factors may be bound at that gene and various other mechanisms to regulate expression of that gene. And the Polycomb specifically affects the K27 trimethylation, which is a silencing mark. So I'm going to color code everything from now on as red in a traffic light system. So if my my nucleosome with my DNA wrapped around has got red. Well, it's got a K27 trimethylation. That's the Polycomb. Polycomb adds that K27 trimethylation mark, and it's reflective of now being in an epigenetically stable silent state through cell division. The opposing mark actually is this K36 trimethylation, so we're going to color code those green. So in a nutshell, what we found was in the plant, the FLC locus, and I'll show you some data in a minute, had the green modification when the gene was on in autumn. And then there was a switch as soon as it felt winter to taking away the green and adding red in a very specific location. And then actually there was a spreading across the body of the gene of this red modification, and you need this very big patch so that when the plants come back into the warm, that patch can be inherited from mother to daughter very efficiently. Because you need to keep that patch. Remember, there's no more cold giving you any more red modifications. You need to inherit it independently of the signal that delivered it in the first place. So if we put that onto our schematic, we have FLCs expressed in the winter here. It's shut down quite quickly, and I'll get back to that in a minute. That's this sensing business. And you have modifications added by our Polycomb complex, which is now a Polycomb plus these two PhD proteins I talked about, which superactivate the Polycomb. So you kick off the K36, and you now add this red mark, the K27 modification, and that's silencing the gene. You see the level here. And it then spreads across the body to keep that gene then off through many cell divisions until you get to the seed. And of course, you have to reset the whole system because you want every generation to need to respond to vernalization. So you then flip the switch the other way in the seed to switch the gene back on again. So it's quite a nice system to study epigenetic switches because it takes a long time to switch it off, so you can really do a nice time series here, but you also have a switch on and a switch off. Just to show you a little bit of data, we map what's going on with chromatin immunoprecipitation. You can buy antibodies that recognize these histone modifications. You grind up your plants, you immunoprecipitate with those antibodies, and then you inspect for enrichment of the DNA sequence around the target or break gene. So with increasing amounts of cold, and this is right at the end of the cold, 2 weeks cold, 4 weeks, 6 weeks, 8 weeks, what you can see nicely is that there's this quantitative increase in the amount of the modification with increasing cold. So that nicely explains why we had this quantitative silencing. When you bring the plants out into the warm, and this is 7 days after transfer into the warm, you then have spreading of that silencing mark across the body. I should show you that the gene is down here. This localized nucleation during the cold is actually just downstream of the promoter, what we call the nucleation region, and that's enough to silence it in the cold. But to keep it off in the warm, you have to spread the K27 trimethylation over the body of the gene. Now, at this point, it's like, okay, we have a quantitative silencing, we have quantitative changes, done and dusted. But actually, someone said to me at that point, well, Caroline, is that a quantitative increase at each gene, more and more and more? It's like icing on a cake, thicker and thicker icing. Or is it that each cell is actually a switch, and you're looking at a population average, which, of course, when you grind up lots of cells, are indistinguishable? And actually, we spent a lot of time since then looking at that. And actually, it's a population average. It's not that just there's more and more and more K27 at each locus. Each locus is either red or it's green, K27, K36. And that's one of the reasons why it takes so long for vernalization to occur. And of course, it needs to take a long time. The plants need to make sure they've had the whole of winter rather than just some cold snap in autumn. So each cell makes that decision. So we've schematically shown that here. So blue, the gene is on. White, the gene is off. So each cell, and in fact, each locus, makes that decision independently. And it's a very low probability switch, so it takes a long time for all the cells to switch off. But as you can see, if you grind these up, it would look like a nice quantitative change. And that's something, as molecular biologists, we really ought to ask ourselves more often. Is this quantitative effect I'm seeing really quantitative, at the particular gene we're looking at, or the particular enzyme reaction we're looking at, or is it a population average? Okay, so I'm going to leave the remembering bit there, and I'm going to now switch to sensing. So I'm sort of working backwards up the system. So sensing winter, and this is more of the recent work, in that how does a plant monitor these complex fluctuating temperatures? What I've shown you so far is we've got this nucleation of the Polycomb complex in the middle of the gene, and then the spreading. And I didn't really make a meal of it, but VIN3 is the only one that's cold-induced. And we looked at all of our factors. Everything was there, quite happily in the warm, but what was switched on during the cold was the expression of this one of the cofactors of this PRC2 complex. So we thought, okay, well, so that's nice. We've got sort of a cofactor which is going to help make the whole system cold-dependent. But when we looked at FLC downregulation in a mutant now completely missing that VIN3, well, in fact, we had completely lost the memory. The gene switched back on again instead of staying off in the warm. But the early phases really look quite wild-type. So we realized that there must be other cold steps involved in the process. And that's what took us into this world of non-coding RNAs. We looked at a whole range of things in a very non-biased way. What else changed in the cold that quantitatively correlated with the silencing of FLC? And, we did this some time ago. I think we published this in 2009. In those days, non-coding RNAs were not really the flavor of the month. Now, actually, lots of epigenetically regulated genes seem to have non-coding RNAs associated with them. So it's quite a common thing now. What we found was if this is our break gene here, here's this nucleation region just downstream of the start site. These are the exons. This is the normal transcript for the spliced sense transcript. At the poly A site of our sense transcript, there was a promoter for a series of antisense transcripts. Now, when we first published this, people were like, oh, well, antisense transcripts, no one really sees much of those. And we didn't, based with the techniques that most people were looking at. But now the deep sequencing has become so deep. People are now seeing antisense and non-coding transcripts for most genes. They're just very low abundance. They're turned over very rapidly because they have nonsense codons in them and what have you. So this was always seen as a bit of an oddity, FLC is now becoming much more of a sort of canonical how to regulate genes via chromatin. And so, look out for non-coding transcripts at your gene. They're hard to see, but that's just because they're turned over rapidly. Anyway, at FLC, you have a non-spliced version, and then you have a spliced and polyadenylated version. And so we've published over many years. There's lots of regulation via this. But I'm just going to focus on the cold bit today. So the question is, do they make any difference at all to vernalization? So we generated an FLC gene now with a different 3' end, so that it couldn't make any of these antisense transcripts. So when we then make a whole series of transgenics and pool them, this is now the reduction of FLC in the cold with increasing weeks of cold with no antisense. And my postdoc at the time thought it was hilarious to call these transcripts cool air, because there's a set of genes in the animal homeobox silencing called hot air. So we have hot air and cool air. So this is minus cool air, and this is the wild type with cool air. And you can see there's a very big difference in how fast the gene is transcriptionally shut down during the cold. So what we see now is that we think we've got this downregulation here of the transcription because of the upregulation of antisense transcripts. And that's an important first phase, and you actually need to shut the transcription of the gene down to be able to nucleate your Polycomb silencing efficiently. So once you're transcriptionally silent, then you can nucleate and you silence, as I showed you, digitally like this in a cell-autonomous manner, and then you can remember that Polycomb off state through many, many cell divisions because you've spread that complex across the whole locus. And then you switch back on. Now, we wanted to have a look, and because this was a switch here, we wanted to understand, so what was going on at the level of the RNA? When you grind all the plants up, as I said to you, and you see quantitative changes, you should be saying, is this quantitative because it's quantitative in each cell or because it's a population average? And, of course, it was hard to actually look at RNA in individual cells. There had been a technique developed in I think it was initially in Cerevisiae, but then had used in C. elegans in the worms, where they used single molecule fluorescence in situ hybridization, putting many different oligos with flaws, so you can look at the oligos down the microscope, and then getting those into the cell, and they find the specific messenger RNA, and then you can look in the microscope at individual messenger RNAs. So it had never been done in plants, and I sort of, this is the joy of being a project leader now. You go to your group meeting and you say, I had this great talk, and they're looking at single molecule RNAs, and if we could just do that in plants, then we could understand at the cellular level, are we seeing the digital effect at the RNA level, at the antisense level as well? So actually, a student, Susan Duncan, took that on, helped by Stephanie Rosa. They got this to work, and it's just amazing. So here is a plant cell. Now, this is one cell with the nucleus counterstained with DAPI. Here's our gene. This is the gene, the schematic you've seen. And you can actually label both strands. So green here is now exonic probes. You have to have about 30 probes, so you have to have a big enough patch to see the sense transcripts. And we can also make probes designed to the intron here. So when you look at green, these are single molecules of messenger RNA in a plant cell. I mean, you can, you really start, you actually count them now. You can quantify exactly how many molecules you're working with. And within the nucleus, what you can see is here are two red ones. This one's yellow because there's a yellow, red and a green on top of each other. These are the sites of transcription. So you're looking at the gene and the nascent transcripts coming off that gene. This opens up a lot of possibilities. So they got this to work. This was for the sense transcript, which of course was more abundant. So the next challenge was, well, can we see the antisense transcripts as well? And yes, indeed, you can see the antisense transcripts. We can only actually see the nascent, the unspliced form, which is interesting in itself, and we make it to this large intron here. And before the cold, and we look in roots because it's easier to visualize, you haven't got the chlorophyll background, but you also have these beautiful files of cells where you can then look at many different cells at the same time. And most of the cells in plants with no cold show absolutely no expression at all. It happens to be one where two adjacent cells are expressing the nascent antisense. But as soon as the plants have sniffed any cold, here we've got 2 weeks, then in most nuclei, you see expression of the antisense. So it's a sort of cellular on and off. And when you then look at a time series, and I've just cut now the nucleus out so I can fit a lot on one slide. So there's a small number of cells make the Coullard transcript over the gene. And these are very larger. So there's more than one molecule of RNA at the gene, which is quite interesting. And the first thing that happens in the cold is these clouds, as we call them now, get even bigger. So it's like the antisense is being made and then kept at the locus. And what's interesting is I showed you the cat, the X inactivation. The exact same thing happens on the X chromosome. A non-coding transcript is made and coats the X chromosome, exist RNA. So I think this making non-coding transcripts and keeping it localized in cis at the chromosome is going to be an important feature of these epigenetic silent states. So that's after 1 week cold. 2 weeks, they start to blib off, and you get these big clouds dissolving into multiple clouds, and then it's a transient thing. So by 3 weeks, it looks like non-V again. That is actually at the locus. We managed to do the DNA RNA fish. And this is all published last year. And then when we look at the same nucleus at both sense and antisense, we can put both probes on the same nucleus. What you see is that you only get expression of one strand at any one time. And that was really important. So you either see an antisense or a sense, but you never see it at the same locus. Very occasionally, you see it in the same cell. The two alleles are expressing the different strands, but you never see a yellow one in the nucleus. So that is saying, and this is very consistent now with a lot of work coming out of yeast, as a gene, when you grind things up, you see sense and antisense. But that's because you're doing a population average. At each gene, at each time point, you make one strand or the other. And of course, it makes perfect sense, because if you want polymerases coming along, you don't want them crashing into each other. So you coordinate which strand is being transcribed. So far, what have we got? Well, we've got our various complexities of antisense here determining the expression, we see a transient increase of this antisense expression, which shuts down the sense expression, and that then allows the nucleation of the polycomb silencing and then the spreading, which keeps you epigenetically silent through the whole of the development. Now this switch back on again, we've actually cloned one of the genes. It turns out to be a demethylase of this particular modification, so it looks like a reversal of the whole process. Now I just want to end with a little bit more about more of the natural variation work, because it basically, that third question that I was telling you about, well, how do plants adapt to different winters, turns out to be very subtle tweaks of this mechanism. So when we looked at natural variation in Arabidopsis, and of course Arabidopsis has a very big geographical range. It's sort of up to the Arctic Circle and down to the Cape Verde Islands. So, you know, they really have adapted to a wide range of winters through lots of different strategies. But again, most of it seems to come down to regulation of this single gene somehow. Now, thinking specifically about the winter aspect, we then collected a whole series of accessions and gave them weeks of cold. So, you know, here, your normal lab one would need 4 weeks. So some of these need really long periods of cold, some of them up to sort of 14 weeks to fully saturate what's going on. But you then see an acceleration of flowering, a decrease in leaf number, and a decrease in FLC level. But they're very characteristic and different from each other. So this orange one, which I'll tell you about, was from northern Sweden, Lurvik. You could give the plant 4 weeks of cold. It was as if it hadn't noticed. It was used to very much longer winters. This was a cold snap in autumn if you're a northern Swedish accession. And then it did respond. So we did a lot of analysis and QTL analysis and sort of did lots of genetics. We could have ended up studying lots of different genes, but no, most of the QTL mapping took us back to the same gene, which, you know, is very happy for us because we only need to think about one gene. When we then look by sequence at all these accessions, what we find is the FLC gene, this is about 10 kb of FLC sequence, falls very distinctly into different groups, haplogroups. And the phenotypic analysis that I showed you, we'd managed to just pick different ones here based on kind of different signatures of vernalization response. Turned out we'd picked one of each of these groups, which said to me that whatever the difference was here, that was the underlying basis of that phenotypic variation. And this was all non-coding cis variation. The amino acids were all exactly the same. The protein function was the same. So it's regulation that was different. Now, when we did this, people were a bit skeptical, I have to say. Like, oh, are you sure about this, Caroline? You know, it's really, evolution isn't so simple as that normally. So to really show that a lot of the variation was at FLC, and that was important, we then cloned each of these out and put them into a common background, and then did that vernalization experiment and compared it now to the behavior in its natural accession. And this is really showing that, you know, the R-squared is very high. Most of the variation that is responsible for the accessions are captured in 10 kb of genomic sequence that we put into our transgenic. So the non-coding SNPs are the key to this difference of vernalization across our different geographical regions. So when we then looked at more detail at one of them, I'm just going to end now with this work on Lurvik, this northern Swedish one. If this is our normal lab one, a Columbia with a vernalizing Columbia, 4 weeks is enough to fully silence the gene. 4 weeks, as I said, was just absolutely not enough for this northern Swedish type. The gene switched back on. But if you give it 12 weeks, then it's fully epigenetically silent. And it turns out there's just 4 SNPs which make all the difference to this ability of need, or the requirement for 4 to 12 weeks. So 4 SNPs isn't very much. So these things, you know, people often say to me, oh, climate change, is it going to, are our plants going to adapt quickly? Well, if you only need 4 changes, then, you know, potentially they could adapt very quickly indeed in terms of changed vernalization. Now, there was no obvious, really strong distribution of those haplotypes, those different FLC groups with geography. So the current work at the moment is to go in and say, okay, why are these different groups, and what benefit are they conferring? And we've managed to, because we sort of quite accidentally managed to pick quite a lot of our accessions, happened to be from Sweden, that we'd done a lot of the molecular analysis, we thought, Well, we really need to go in and understand what adaptive changes, what adaptive benefits those SNPs might be in the natural location that these plants have been growing in. So this Lervic one is right from this region in the North Sweden up here in the high coast, and then the others we've been looking at are down here from the south. And so we've had field experiments for the last 4 years in the north and in the south, and we're incredibly grateful to Svante Holm from Mid-Sweden University, who helped us with the north plot, and Torbjörn Sahl from Lund from the southern plot. And we really just want to understand, what's the benefit of these SNPs and how is the mechanism influencing the adaptation. So I get to go to nice places. So actually it is quite nice in northern Sweden if you go at the right time. So this is the Lervik site, absolutely beautiful. It's a part of the world that it's still rising quite rapidly out of the water because the ice was so heavy, it's still going up. So these islands keep appearing out of the water. The students, however, have a much tougher job. Every week they're going out, digging up the Arabidopsis, which we've planted, and sending it back to Norwich. So, this is, poor girl, 3 feet of snow. The plants actually are absolutely fine. But now we've got these, we're trying to understand, when is vernalization happening in these very natural conditions? So in this particular one, here the plants were fully vernalized. This was in the end of November. In this Lervic type, which was surprising because if you'd looked at this temperature profile, you'd said, well, this nice, stable, very low temperature would be when the plants would detect the winter. But actually, that's under the snow, which is why the temperature is so stable. And in fact, they're fully vernalized before that. So the plants are somehow integrating this very complex profile. So this is one tiny snapshot of a big project where we've got all this stuff, lots of accessions all sown out in Norwich as well, so we can look at transgenic lines, and we're trying to really understand what are the genetic requirements that are important for monitoring that fluctuating temperature and integrating it into modulating this epigenetic silencing of FLC. So here's the temperature profile. You see, actually, the field stuff is a bit of a nightmare to work with, but actually it works reasonably well in terms of sending the material back and processing. These are all different genotypes, and actually, they look like they behave really rather similarly, except for one batch. And here's our Polycomb batch and our VIN3 batch here. So these have completely lost the ability to vernalize. Anyway, we're in the middle of a big analysis, so that's what's ongoing in the lab. And it's a really important analysis because, winter is changing, and the fluctuations in winter are really changing. And one of the biggest, I think, surprises has been that the plant seems to be, from the modeling of that data I've just shown you, not that it's monitoring some average temperature, but it's monitoring the fluctuations, the highest and the lowest points. And of course, those are probably the ones that are going to change most with climate change scenario. So yes, we need to do this. We need to do this for agriculture as well as actually understanding vernalization. But the one really thing that has amazed me is just how much we've learned about basic epigenetic processes from studying this one gene in this plant, that people now across the broader epigenetics field, mammals and everything, are really using this gene to see how all these different aspects integrate together. And so I just thought, well, I'd put vernalization, these are the generic lessons that people, if they're working in the non-plant field, actually follow and understand. That this quantitativeness is this digital silencing. And this is now catching on big time in the whole field. The memory is held in the local chromatin. I didn't really get into this, but it's not through transfactors. Each locus switches and the memory is held at that local chromatin. And that's true for switching and for memory. It's an interesting timing mechanism, because if you think of cells, they've got to somehow perceive signals, whether it's a developmental signal, whether it's a signal from a pathogen in the soil or anything. And they're waiting and waiting, and they think, have I had enough of whatever I'm looking for? And then they make a decision. So this kind of monitoring and epigenetic kind of fixing of, yes, I've had enough, but quantitatively, so you can kind of do a quantitative averaging, I think is going to be a concept that's very generic across many different biological traits. We've got lots of aspects of non-coding RNAs, so the COOL is becoming very famous. And of course, in the adaptation, really just a few SNPs make a very big phenotypic difference. And so in terms of evolutionary field, this is quite interesting. So I've given you a kind of really high-level overview of sort of what we've been doing and what we're currently doing. And happy to talk anyone later about any of the specifics, but I hope I've managed to explain to you sort of the basics of Polycomb and epigenetics, if nothing else. And it just leaves me to thank my team. Of course, I've talked about work over many, many years. This happens to be at the field site that a year ago with the various people, and all of their nationalities. All the Swedish people here, of course, live in Sweden, but all of the other people have been people in my lab, and that's a good thing. So I'll acknowledge them, and I'll take questions. Thank you.